Hello, this is Professor White, and we are on our second lab here in our Bio 102 for your laboratory in biology. So let me share my screen real quick. So this is about biomolecules or biological molecules. So the objective of this lab is for us to actually write a hypothesis statement based on knowledge of biological principle. Last week, on our first week, we explored what a scientific method is. In fact, during our discussion, we did mention some real world examples of how we use scientific method. So we are going to practice that as one of our learning objectives in the lab. We're also going to identify experimental variables such as independent, dependent, and controlled variables. Also, we're going to distinguish between quantitative and qualitative data, as well as determining the presence of different biological molecules like amino acids, proteins, glucose, starch, using indicator solutions. Also, we're going to discuss the different biological interaction based on the chemical test reagents that we will be using in the lab, as well as discussing the functional aspects of different chemical functional groups in biological molecules. And last but not least, to recognize and identify the chemical functional groups in biological molecules. So I'll start first with the gist of the activity, especially on the questions that you guys will answer for the lab, which is conducting experiments. So all throughout the course, you guys will be conducting experiments. And so experiments are the foundation of science knowledge. And we carefully design these experiments to assure that the results that we get are clearly interpreted and, of course, accurate and precise. Now, all variables are very precisely defined, measured, and controlled. And the first step, of course, of an experiment is to come up with a question based on an observation. So that's conducting experiments in a nutshell. We'll start first with independent variables. So just to make it very straightforward, independent variables are the variables that we actually manipulate or what we change. Now you set the value of that. It could be different amounts of concentration or the amount of nitrogen in the soil, for example, for those some of us who might like gardening, or the amount of water that you put in your plants. So that is an independent variable. Now, dependent variable, on the other hand, is the measurable outcome depending on the value of the independent variable. So if, let's say, for example, you change the amount of nitrogen that you put into your plants, then the outcome or the measurable outcome could be maybe the number of tomatoes in the vine after a period of time. So that is a dependent variable. So this is the main experimental result. So that is a dependent variable. Now, how about controlled variable? So when you say controlled variable, these are the variables that was not changed at all. Constant variable. So this do not impact either the independent or dependent variable. So what are some examples? The very practical one from, you know, planting perspective is the same amount of sunlight or the same amount of water that you give the plants if you are using this in the experiment. Okay, any amount of water that plant setup A has is the same with plant setup B, and that is called controlled variable. And of course, if there are changes that you did to your setup, there's of course your experimental results. It can be qualitative or quantitative. So quantitative basically is in a form of a value or a number, for example, count of tomatoes or the height, or if you count number of pieces, for example, or the time, for example. These are all quantitative. Now qualitative, on the other hand, these has to do with what you observe with your eyes, with your senses. Let's say, for example, the appearance of blotches on tomatoes, for example. So these are all qualitative um, qualitative data. Okay, so that's basically it. So for our lab this week, we are dealing with biological molecules. Now, all matter is made of atoms, and thus living beings are also made of atoms as we speak. That's why it's the chemistry of life. So chemistry is basically very important in biology. So when we look at our periodic table, last week I posted a video about a little review about the chemistry of life. We know that the periodic table have different elements, and these elements can form molecules, can form substances, right? And then they form molecules, right? And one of those kinds of molecules that we are going to focus on is the biological molecules. And the interaction of the elements form the basis of living organisms. And that's your biological molecules that we are talking about. And all our organelles, our cells, tissues, organs, and the whole body is composed of these different biological molecules. And they arrange themselves into what we call functional group. So this diagram right here is called your functional groups. As you can see here on the image of this biological molecule or biomolecule, in short, it is represented by amino acid, carboxyl group, and there's what we call this R group. R represents a group of atoms attached to the functional group. The R group is referred to as the side chain as well. So that's your biological molecules. And one biological molecule is your carbohydrates. So carbohydrates, aka these are your sugars, your saccharides. They have a hydroxyl functional groups and aldehyde functional groups. And these aldehyde group of saccharides interact with chemicals in a test solution called Benedict reagent. So in the lab, you will use this reagent to test for carbohydrates. Now, this test result in a change in color when a specific kind of sugar is present. Okay, so that's called a reducing sugar. Now, monosaccharides such as glucose or simple sugar, fructose are reducing sugars, and you'll see that in the lab. Now, the next one is your proteins. Now, your proteins are composed of polymers of amino acids called peptides. Now, there can be many amino functional groups in proteins, and these groups interact with chemicals in a test solution called Bire test reagent. Now, note that some peptides functions as long strand of molecules rather than proteins, and these amino acid and long strands can also interact with the BRA test solution. Now, nutritionally speaking, we categorize macromolecules as sugars, lipids, which you'll see in the test also. It's not mentioned here on this slide, but Sudan 4 test is actually the test reagent for the presence of fat. Now, please note Benedict and BRA testing solutions are mentioned as just examples. And you'll see in the actual lab, you will in fact use entirely different testing solution for the lab today. So for procedure one overview, you are going to have the known sample solutions, and you're going to observe the reaction or color change when different test solutions are added to the set of known sample solution. And then for the procedure two, you have unknown sample, and you're going to observe the reaction or color change when different test solutions are added to a set of unknown sample solution. Okay, so just to share with you the lab paper, as you can see over here, please make sure you don't forget to screenshot. It's a part of the instruction, so please read the procedures. Okay, so you will have this activity form right here that you have, and you are going to fill in and complete the table for each sample solution. You're going to indicate any color change that occurs when the test solution is added. So when you run the simulation, fill it in, and you're going to find out the macromolecule classification of each. So if I will, let me go back to here. Let me stop my sharing for that one real quick, so I can go to the lab itself. You have the procedure on here. Please read them. As you can see, at some point it asks you to take a screenshot on the procedure, so please don't forget to attach that onto your assignment. Okay, so the activity will let you run the procedure, follow along the procedure. That's very important for you to get answer for your table. Okay, so the table, if I did not mention that, it's half a point every box for it when I check it. And then when you go to the next page of your paper, you'll have those questions. And the questions are actually linked to your scientific method. So, write a hypothesis, a statement for the experiment you conducted, and stuff like that. So, when you formulate your hypothesis, that must be aligned to the goal of the experiment. So that's basically what it is that you will write your hypothesis on. Also, it is helpful for you to look here too, and that we know that the group 1 sample solution, A, B, C, D, E, A is sugar, starch, fat, protein. So please make sure that you know this. Okay. So I just wanted you guys to know about that. So as you go through the questions, please read it. Make sure that the dependent independent variable is clear for you, and then qualitative, quantitative when you answer the questions. Okay. So it says over here, test one solution reacts with, complete the sentence and all that. So that's your next one. And then when you go to procedure 2, same thing, but this time it's the unknown sample solution. Okay. And then again, there are questions that follow. So please read the question and make sure that when you write your answer, they are clear and they're in a complete sentence so that I know what you mean and it's clear if I read it. Okay. So follow along until you get to the last page, which asks about the second part of the lab. So if you have any questions, please send me an email. Please use our school email, not the messenger on our learning platform. Please make sure that you get in touch with me if you do have any questions, but it should be very, very easy. Other than that, have a great day, and I'll see you in class. Bye.